Welcome friends. Today we will be discussing with specimen stains. Now this is very interesting and very enjoyable practical to do in the lab. You must have done it, I am very sure. So you must have enjoyed it also. So today we will be discussing the principles of specimen staining. Okay, we will be studying about the theoretical knowledge of it. You can relate it to what you have done in the lab. Okay, so see. So what happens? The specimens which we use, they are colorless. To observe the different contents of a cell, what we need to do? We need to sometimes we need to stain the different components of the cell with different color. Okay, isn't it very interesting, guys? See, the contrast can be introduced into the specimen by using colored dyes or stains. The stains can be non-specific stains, which say for example a general protein stain such as Comasi blue. So here we will be discussing about non-specific stains and specific stains. So one example of non-specific stain we discussed, what is that? It is Comasi blue. These all are very important for MCQ questions, guys. Please note it down. Okay, or a stain which can label an organelle. For example, the stains which can label an organelle, say for example nucleus, mitochondria, they can give different colors to different organelles. Now you will be knowing that how the stain will know that which organelle has to get which color. Okay, we will discuss about that also. And these contrasting colors which are added to the cell, these histological stains, which are histological stains here means they are giving colors to the different parts of the cells. And they can be observed using bright field imaging or bright field microscopy. Clear? Now, viewing these contrasting images, it's not limited to the light microscopy, to the bright field microscopy. Instead, other light microscopic technique can also be used to view the entire tissue along with the stained tissue. For example, differential interference contrast microscopy can be used to view the entire morphology of an embryo and a colored stain. Clear? Again, more specific dyes are usually used in conjunction with the fluorescence microscopy. For example, immunofluorescence microscopy, it is used to map the spatial distribution of macromolecules in the cell. Where are the macromolecules located in the cell? And this method of epifluorescence microscopy, they require the highly specific binding of antibodies to the proteins. So here we write that specific stains we use mainly in fluorescence microscopy. For example, mainly in immunofluorescence microscopy, where we need to find out the spatial distribution of macromolecules in the cell. So first, it will be better if I discuss with you what is immunofluorescence. This is a main topic in immunology, but as the term came here, we need to have a brief idea about that. So immunofluorescence is an assay. We are discussing about the term immunofluorescence. What is immunofluorescence? This immunofluorescence, it is an assay which is used primarily on biological samples, and it is defined as a procedure to detect antigens using antibodies. So I can write here what is this immunofluorescence. It is an assay used to detect antigens using antibodies. Okay, The specificity of the antibodies to these antigens, this is the base of immunofluorescence. Specific antibody will bind to the specific antigens. Clear? This is immunofluorescence. Now, what is fluorescence? You already know. Clear? What is fluorescence? If you don't know, that there is a property present in certain dyes which absorb the light at particular wavelength. Say for example, they absorb ultraviolet light and emit them at a different wavelength. Say for example, visible light. All the time it's not visible light, but say for example, it is emitting in the visible light, and this is known as fluorescence. Now, in immunofluorescent test, the fluorescent dye which illuminates in the UV light, that means it is excited in the UV light, are used to detect or show the specific combination of an antigen or antibody. That means the antibodies here are labeled with the fluorescent dye, and if they bind with the specific antigen present, then these antibodies which has the fluorescence molecule attached, they will emit light. Clear? So see here, we need to talk about the indirect immunofluorescence and the direct fluorescence. So here see, antibodies, I told, these antibodies are raised to the protein of interest, means they should bind with the specific antigen, and here protein of interest means they are going to bind the antigen which is we want to detect. Okay, they are raised to the protein of interest, and these antibodies are labeled with a fluorescent probe. Okay, and this probe is used to label the protein of interest in the cell. If the protein of interest in the cell is present, then this antibody will bind to that protein of interest and it will emit light. Okay, once it emits light, it can be imaged using fluorescence microscopy. And in practice, cells are usually labeled using indirect immunofluorescence. Very important to know that in practice, cells are usually labeled with indirect immunofluorescence. Now, what happens in indirect immunofluorescence? That in indirect immunofluorescence, here it is double antibody technique. So, I write here this indirect immunofluorescent technique. This is a double antibody technique. Very important. Because this is similar to immunology also and microscopy also. So, two topics are covered when you do this chapter. Okay. Double antibody technique. Here, the unlabeled antibodies which have bound to the antigens, say for example, these are antigens. Okay, I use different color for antibody. Say for, okay, all color changed. I cannot use different color. Black would be fine. Okay, black would be fine. So, these antigens are binding to the antibody. Okay, they are binding to an antibody. So these antibodies are not labeled at first. The unlabeled antibodies, they are binding to the unlabeled antibodies. Now, the unlabeled antibodies which have bound to the antigens are visualized by a fluorescence anti-globulin reagent directed at the unlabeled antibodies. So, directed at the unlabeled antibodies are another antibodies which have been labeled with the fluorescent. Okay. So, you can see here that these are antigens. This is virus specific unlabeled antigen, and another one is the fluorescent labeled, if I said antigens, antibody, it is virus specific unlabeled antibody. Okay, the second one is virus specific unlabeled antibody, and the third one is this unlabeled antibody specific. Unlabeled antiglobulin, the term we use. This is unlabeled antiglobulin specific. Clear? Is it going into your mind what I said? Fluorescently labeled antibody. Okay, so I write here the first one is the antigen or the protein of interest. To the antigen, what is binding? It is binding unlabeled antibody. To the unlabeled antibody, what is binding? It is binding fluorescent antiglobulin. Fluorescent antiglobulin. This fluorescent antiglobulin will give the color. And this indirect immunofluorescence test, it is used for the detection of specific antibodies. It is used for the detection of specific antibodies. I write here the uses. It is used for the detection of specific antibodies in the serum and other body fluids. Okay, for serodiagnosis of many infectious diseases. Okay, many infectious disease can be diagnosed with this method. I hope it is clear. The more detail of this topic we will be understanding when any day we will be doing with immunology, or you must be knowing it before if you have studied immunology. So this was about the indirect immunofluorescence microscopy. Now, what is the need of using the fluorescent molecule tagged with the second antibody? Why? Because such a method or such a protocol gives higher fluorescent signal than using single fluorescent labeled antibody. Very important. In the interview, you may be asked that what is the use, like why indirect immunofluorescence is more preferred than the direct immunofluorescence, because just remember the difference is in the direct one, the fluorescence is added to the first antibody. Okay, there is no secondary antibody, there is primary antibody only, and the fluorescence is added to the primary antibody. But in indirect one, the fluorescence is added to the secondary antibody. And why is that so? Why is indirect immunofluorescence more preferred? Because it gives the higher fluorescent signal than using the single fluorescent label antibody. Got it? So there is some procedure which has to be followed in this immunofluorescence protocol. What is that? First, fix in 1% formaldehyde for 30 minutes, rinse in cold buffer, block the buffer, incubate in primary antibody, example mouse antitubulin, wash 4 times in buffer, then incubate in secondary antibody, example secondary antibody I told you have a fluorescence molecule labeled with it. So fluorescence labeled rabbit anti-mouse, wash 4 times in buffer, incubate in anti-fade reagent, example Vecta-Shield, then mount on the slide with a cover slip and view using epifluorescence microscopy. This is the general protocol. So here the first important step which includes is that incubation with the incubation of antigen plus primary antibody. Here the primary antibody is unlabeled one. Okay, and here what we see, after that there is a careful washing step. Then after that we see that there will be the incubation with the secondary antibody. Here the primary antibody will bind with the secondary antibody. Okay, and this secondary antibody have the fluorophore attached to it. Clear? And primary antibody, if it's still attached with the antigen. Okay, understood? So that was all about immunofluorescence microscopy.